My name is Helen, and today we're talking about how to document changes that are occurring to a medical device and how to assess their effect to the overall biocompatibility of the medical device. A change in the biocompatibility world is considered an alteration of a medical device that stems from either a change in the materials that are used or processes, a change in a site or location of the manufacturing process, any changes occurring to the supply chain, packaging, sterilization, and so forth. As any of these changes could potentially affect the biocompatibility of your medical device, it's very important to evaluate the potential risks that are introduced to the medical device when it's undergoing these changes. It is very common to run into this problem when we have a medical device, let's say this pen, and it has already undergone all of the biocompatibility testing or evaluation, and now we want to change let's say a material in one of its components, or add a colorant for easier size recognition, or maybe change something in the manufacturing steps. Does this mean that we have to go all the way back to square one and start testing all over again? The short answer is no, or to be more accurate, not always. As I said, potentially any changes that are occurring to the medical device could affect its biocompatibility. But we always have to look at the risks associated with the change from a scientific perspective. Luckily, we have the support of the new ISO 10993-1 that came out last year in 2018 that strongly emphasizes the use of a risk-based approach when assessing biocompatibility or changes occurring to the medical device. We also need to consider the intended use of the device based on its patient contact site and duration, and what specific risks are associated with that specific contact. Intended use is key here. When we want to evaluate how a change affects the biocompatibility of a medical device, we have to go and look at the risks that are associated with that specific part of the device. Basically, we have to go back to the table A1 in ISO 10993-1 and categorize that part of the device that is undergoing the change based on its patient contact. And I did say that part of the device because not all parts are created equal. Equal in the sense of what risks they carry in terms of patient safety. Let me give you an example. Let's say the cap of the pen is undergoing a change. Let's say we're wanting to add a colorant to it. So this cap is going to be removed from the device prior to its use on a patient. We also know that the cap is designed in a way to minimize contact between the cap and the tip of the pen that has the patient contact. So based on this background knowledge that we have on the cap, this change really does not affect the patient, and the risks associated with this change are thus minimal. Now, if the tip of the pen was undergoing a change, let's say a stainless steel tip is going to be changed to a more flexible polymer, what are the risks there? First, we have to identify what is the patient contact that that tip is going to have during the intended use of the device. Let's say this pen is going to be used as a surgical marker to mark the incision sites for surgery. And let's say it's going to be used up to 4 hours. Now, this tip will have then, based on the categorization of ISO 10993-1, limited contact to intact skin. And based on the table A1, the specific endpoints that need to be evaluated for are cytotoxicity, sensitization, and irritation. Now, if this tip were to, during clinical use, contact normally sterile tissue, let's say in case of a laparoscopic instrument, the categorization would be different, and the endpoints for evaluation would also be different. And the risk associated with that change would then be higher. Now, after we have identified all of the potential risks that are associated with this change, we want to know everything there is to know about that change and how that change actually affects the biocompatibility of the medical device. For instance, if there is already some biocompatibility information available, let's say on the material that is being introduced to the medical device, then great, we can absolutely use that. This information can be found either in material data sheets, internal testing reports, or published literature. Also, oftentimes, it's a good idea to reach out to the supplier of the new resin or the material and see if they have any biocompatibility related information already at hand that they would be willing to share. Then that could absolutely be used as well. It also should be considered how the manufacturing steps that you are using to produce your device, let's say cutting or machining or molding or sterilization, could affect that new material, to make sure that those steps are not altering the material in any way. I want to emphasize that in the risk assessment, all of the previously identified biological endpoints and risks need to be evaluated. If a scientific rationale can be provided to mitigate all of these risks, then great, no further testing is required. If some, but not all of the endpoints can be mitigated using a written justification, then a limited testing plan may be recommended, and so on. It is very important to remember that if anything is changing, anything at all in your medical device, it has to be documented. And also, the documentation should be clear and the rationale provided within easy to follow so that the reviewer can understand the trend of thought. Therefore, regardless of the change, it should be evaluated in written documentation that could be provided upon a request by your boss, your colleague, an auditor, or even FDA, or whoever is wanting to see your biocompatibility files. I hope it was helpful. Don't hesitate to reach out if you have any questions or if you want any assistance in specific projects that you have.